Transcription and Translation, how our genes code for our traits.
So did you ever wonder why you're as ugly as your dad? Well, the basic reason for that is because he gave you some genes that gave you some traits. That is, some DNA that caused you to have certain physical characteristics. But this is oversimplifying things, because to get from genes to traits requires several other steps in between. Specifically, when we say genes, we're really talking about the molecule that contains the code for those genes, specifically DNA. And when we talk about traits, we're also talking about molecules inside our bodies, this time proteins. Proteins are the molecules that occur inside our cells and that cause our cells, and thus ourselves, to look a particular way. And in order for our cells to read the DNA and use the code in the DNA to make proteins, they need to make an extra molecule in between, and that's the middle step. This molecule is called RNA. This series of steps is the focus of this lesson: how our bodies use the code in our DNA, turn it into RNA, and use that code in the RNA to make our proteins, which then cause us to look a particular way. So in other words, in order to get from our genes to our traits, we really need to take steps through these three molecules. Here's an artist's interpretation of what this might look like inside our cells. You can see a cell membrane that's cutting through this picture this way and off through the back. This is a picture of a cell. And if we were to zoom in on this cell, specifically over here on the nucleus, we could see a long strand of DNA inside this nucleus that's being pulled apart into two strands right here and being read by this large enzyme. And the enzyme is copying that DNA, making this long molecule snaking off to the right. What is that? That is the RNA molecule. And so you can see this RNA molecule over in this area of the cell being read by this long molecule. And this reading molecule is not only reading the RNA, it's also using it in order to make a new molecule. Finally, the protein. You can see the artist's long interpretation of the curled up protein right here. And if we were to zoom in on the protein, we'd see the small amino acids that make up this molecule. So overall inside this picture, we're seeing this transition. Again, code in the DNA being copied into RNA and then being read in order to make proteins. And this is what's going on inside our cells in order to get from our genes to our traits. One analogy we've discussed before is, let's pretend that the nucleus inside each of our cells, this being one of our cells right here, is a little castle. If we zoomed in on the castle, right, let's take a quick peek. Inside the castle, if we looked inside the tower that's right at the top, we would find DNA inside. In this case, it's drawn as a little bit of a book. Now, DNA never leaves the nucleus, so how is the nucleus of the cell going to send out messages? That's where our friend the scribe comes in. So remember, inside the nucleus, there is a scribe molecule that's making copies of the DNA, and the copy that he's making is actually an RNA molecule. And when the scribe tosses the RNA out of the window, they go floating around inside the cytoplasm of the cell. Here you can see all of those messages scattering from the nucleus to the cell until they land inside the cytoplasm. Inside the cytoplasm, there are small chefs, which are used to symbolize molecules called ribosomes. You can see this little chef. He's getting the RNA recipe right here. And the RNA recipe is telling him what kind of meal to cook. And in this case, the meal is a protein. In other words, the cell is sending messages out of its nucleus to tell ribosomes how to make proteins, and those proteins are what make our cells look and act in a particular way. The title of this talk included the words transcription and translation. Those two words are involved in this process. As you can see over here on the left, in order to get from DNA to this RNA copy, a process called transcription takes place. And in order to get from the RNA molecule to the protein molecule at the end, a process named translation takes place. These two names are really apt because transcription has this word in the middle of it, script. Script means to copy or scribe, whereas translate, you may already know, means to change languages. And that's exactly what the cell is doing. It's taking a DNA template, so that means inside your genes, a section of your DNA, and it's making a copy of that DNA in the form of RNA. RNA and DNA are very related molecules. Then the cell is reading this messenger RNA molecule and using it to construct a protein. Now, proteins are made not of these nucleotide letters like DNA and RNA, it's made of a different alphabet of amino acids. And so to change from this language to this language, scientists call that translation. So let's start with the first step, transcription. Transcription means copying in the same language. And so when we say that cells read DNA and use it to make RNA, we call that transcription because the languages, i.e. the structures of these two molecules, are so similar. And this chart here compares and contrasts, but mostly compares DNA and RNA and reveals a lot of those similarities, starting with the bottom. And at the bottom of this chart, we can see how similar the nitrogenous bases are therein. Inside DNA and inside RNA, you can find G for guanine, you can find A for adenine, and you can find C for cytosine. The final letters are different, T for thymine inside DNA, and U for uracil inside RNA. But T and U aren't actually that different from each other if you look at their molecular structure. For example, you can see carbons with the same number of bonds in the exact same spaces all over these molecules, nitrogen's in the same spot as well. The only thing that's really different is this small cluster of atoms that's off to the side. So even though thymine and uracil do have different names, for the most part, they are pretty similar. Which just adds to the point. DNA and RNA are in the same basic language, so to change from one to the other is to transcribe, not to translate. DNA and RNA are also very similar in the sugars they contain in their backbones. For example, in DNA, you have this molecule right here, deoxyribose sugar, and in RNA, you have ribose sugar. These molecules are almost entirely identical except for the small cluster of atoms that's over here to the side. You can see that right here in deoxyribose sugar there is an oxygen missing, and that's where the molecule gets its name, deoxyribose, meaning missing an oxygen, whereas ribose sugar has that oxygen. But still, this is just one atom of difference we're talking about. These molecules are still, for the most part, identical. But in the upcoming diagrams, a surefire way to tell the difference between DNA and RNA is to look at how many strands make it up. DNA always comes in this double-stranded structure. That's where it gets its name, double helix, whereas RNA always comes in a single long strand right here. So that's something to keep an eye out for if you're ever wondering if you're looking at a DNA molecule or an RNA molecule. DNA, double-stranded, RNA, single-stranded. When transcription takes place, you can see this difference between the DNA and the RNA. For example, the DNA is over here. Here it is in its double-stranded structure. You can see it, double helix. And then coming out from this structure, you have this one long single-stranded nucleic acid. That's ribonucleic acid, or RNA. Now this diagram is a picture of transcription happening, of the message that's inside the DNA being copied and put into an RNA molecule. And you can see that being done by this large enzyme right here. This enzyme is named very appropriately. It's called RNA polymerase because the A's tells us that it's an enzyme, and what does this enzyme make? It makes RNA polymers, that is those long RNA chains like the one you see right here. This enzyme, by the way, is that scribe making copies of the DNA, those RNA molecules, never forget. And what did the scribe do with those RNA molecules? He tossed them out the windows of his castle. Now the windows of the castle in this case, well here's the castle, it's the actual nucleus that's going around like this, And the windows are these little suckers right here. Those are nuclear pores. Holes in the nuclear envelope that allow the RNA molecules to go out where they will be read. One thing that always impresses me about all this is that scientists never see a pretty cartoon like this when they look inside a cell. Instead, they see something that looks like this. And they look at this picture, and using their vast techniques and years of studying, they're able to look at this and say, all right, this long strand right here is the DNA, this little lump right here must be the RNA polymerase, and sometimes you can see trailing out from the top of it is an mRNA molecule. Very, very difficult to see, so thank you scientists and people who made our textbooks for making clearer cartoons that are easier to understand than this picture through a microscope.